name is Marilyn Palmer, and I'm a rising senior at the High School for Health Professions. And I worked alongside with Yasmir and Arrington, who worked with gram-positive bacteria. But my job was to test the antibacterial activity of mint leaf extraction on gram-negative bacteria. All right, so just a little introduction. As they mentioned, there are 4 common species. Was Piperita, which is also known as peppermint. And it is a hybrid cross between water mint and spearmint. And the oil from the leaf is very popular, especially in other countries. And it is used for things in food products, cosmetics, soaps, and alcoholic liquors. And peppermint oil is known to have antibacterial properties. And some of the healing properties of peppermint oil is to soothe inflammation, respiratory disorders, and weight loss. And like I said, we are researching to test the antibacterial activity on gram-negative bacteria. All right, so our hypothesis was that since gram-negative bacteria has become increasingly resistant to most antibiotics, we believe that Mentha piperita has antibacterial properties to fight gram-negative bacteria. And the 3 types of bacteria that we used were E. coli, Pseudomonas aeruginosa, and Comobacterium violaceum. All right, so a little bit about gram-negative bacteria is that gram-negative bacteria is a group of rod-shaped bacteria that has a single layer of peptidoglycan, and it does not retain the violet color when doing gram staining, and it becomes like a reddish or pinkish color, as you can see. And it causes most of infections such as pneumonia and bloodstream infections. And treating gram-negative bacteria can be difficult because they have a unique cell membrane which is impenetrable, and it prevents certain antibiotics from entering into it. And the difference between gram-negative and gram-positive bacteria is that when doing gram staining, gram-positive bacteria retains the violet color, but as you can see here, but gram-negative does not, and it becomes pink or like red. And gram-negative bacteria has a single layer of peptidoglycan, while gram-positive bacteria has multiple layers. All right, so mint and medicinal practices. In the history of medicine, herbs were used to alleviate certain illnesses and to fix injuries. And mint was used by Egyptians to treat gastric ailments and to stop vomiting. And it was used by ancient Greeks, and they would use it to rub on their foreheads to relieve headaches, and rubbing it on their skin to relieve itching, and inhaling to relieve nasal congestion. All right, so the 3 types of bacteria that I'm using, again, are Pseudomonas aeruginosa, E. coli, and Comobacterium violaceum. And Pseudomonas aeruginosa is a strain of bacteria that is commonly found in the environment, and it causes most of the infections in humans that are resistant to nearly all types of antibiotics. And it causes bloodstream infections and pneumonia in patients with cystic fibrosis. And E. coli is a diverse group of bacteria. Most strains of E. coli are harmless and is found in the healthy human intestinal tract, but it can cause illnesses outside of the tract, and it can be transmitted through contaminated food or water and through contact with humans or animals. And Comobacterium violaceum is a gram-negative bacillus that is an inhabitant of stagnant soil and water, and it can usually start in the skin and progress to swelling in the body and can cause pus. Alright, so the materials and methods that we used were media preparation, the streaking method, mint extraction, and the disc diffusion assay. Alright, so as they stated, we measured 10 grams of agar media. We added 250 milliliters of distilled water. We mixed our ingredients, and after we autoclaved it to sterilize our media. And after letting the media cool, we poured it into a petri dish so it could solidify. And we did the streaking method, as they stated, and we started in our corners, and we turned the plate 90 degrees in order to get a single colony, because single colonies ensures that the bacteria is the most pure. And we did 2 methods of mint extraction, which were the ethanol extraction and the distilled water extraction. And as they stated again, they chopped up the leaves, and we submerged the mint leaves into ethanol. And we did this for 2.5 weeks, and we put it into a dark place because light could have sensitivity effects, and we did this to speed up the process. And the boiling water extraction, we chopped up our mint leaves and put it into boiling water for 100 milliliters of boiling water for 20 minutes. And after letting the water cool, we filtered our mint leaf extraction. All right, and we did the antibacterial sensitivity assay, which is also known as the disc diffusion method. And in Kirby beer testing, the discs contain antibiotics that are placed into an agar plate where our 3 types of bacteria were growing. And the antibiotics diffuse out onto the plate, and the areas around our disc would show how effective the antibiotics were. And these areas around here are called the zones of inhibition. And the larger the zones, the more effective it proves to be. All right, so the testing of our antibacterial activity of the mint leaf extractions, we labeled the agar plates, and after doing that, we inoculated the plates with the 3 types of bacteria, and we placed our sample discs onto the surfaces that had each of our samples, and then we incubated them overnight at 37 degrees Celsius, and afterwards we observed our plates to see if they had zones of inhibition. All right, and our results showed that the water extraction had less activity than the ethanol extraction. And in our experiments, the gram-negative bacteria had less sensitivity to the water and ethanol extraction than the gram-positive bacteria. All right, and this is just a graph showing that the gram-negative bacteria had less activity than the gram-positive bacteria. All right, and so again, our results showed that the water extraction had less antibacterial activity than the ethanol extraction, and we found that the gram-positive bacteria had a greater antibacterial sensitivity compared to gram-negative. And because of the short duration of this program, it's supposed to be 4 to 6 weeks, but we can only do it for 2.5 weeks. So if we have more time, we would do it for more. We would do the extractions for longer. And in the future, we will use other solvents like chloroform and ether and test it against other pathogenic bacteria. And these are my references. And as they stated, we did literature review and gram staining. And I would like to acknowledge Dr. Angela Peters and Burley Tisdale for allowing us to participate. We would like to thank Azima Kalsoum for her great mentoring us with this project. And thanks to Dr. Harrison Panasic for allowing us to use their laboratories. And we would like to thank Ms. Cherry for her assistance and our counselors, Kiana Ware, Edwin Simpson, and Takia McClary. And thank you to Epscor for funding this program. And we would like to thank each of our families and friends for their love and support. And thank you to Claflin University to allowing us to conduct research in their laboratory.
My question is, we've seen your bacteria, we've seen your layers. Now, why, when you look at the layers with the gram-positive and the gram-negative bacteria, you see what has happened, right, as it relates to which one had greater sensitivity. If you were to take that and say you use penicillin or some other type of pharmaceutical, what do you think would happen, what type of effect would you think would happen?
The antibiotics.
No, I'm talking about as far. What has happened is that these organisms are becoming non-sensitive to the current day types of antibiotics that we have. We could probably think of some of these less toxic and also natural products that might do the same effect.
We suggest or other fellows coming after you or other students that those species could be tested to see our difference, how they are different from this species, the one you used.
Yes, other types of species, because maybe they could have a greater effect on gram-negative bacteria. Because as you've seen, the gram-negative bacteria didn't retain it as well as gram-positive, so I think they should test